Good afternoon, everyone. It's my distinct pleasure to introduce Seth Myers today from NanoString. I've worked with Seth and NanoString for the last almost 6 years. If you're not familiar, the SNRI BioCore located over in Stark Neuroscience Research Institute has a nCounter platform with the prep station as well as the GeoMx spatial profiling system. And I think Seth is going to discuss at least the spatial system today and talk about the new technology that's coming online. So I'm going to turn it over to Seth.
Great. Thank you so much, Adrienne. Really appreciate it. Hi to everyone. Great to be here today, this afternoon. I'm very excited to be able to talk with you about some of the new offerings that NanoString has by way of spatial biology. And I think that what you'll find is after today's talk, you'll find that really spatial discoveries, they happen at many scales, right? They can happen at whole transcriptome to spatial single cell, and really whether or not you're looking for discovering new biomarkers or understanding how disease progress or building a cell atlas of tissue, the solutions and really the team here at NanoString are the ones that can help you to reach your discoveries faster. So before I go into some of the technical specs around our instrumentation and the latest we have by way of spatial biology, I just wanted to share a quick webinar kind of recap of what we have that's coming out throughout much of this year. And these are great webinar series that we have that really demonstrate data sets that have been generated with our CosMx spatial molecular imager platform, as well as GeoMx data sets. So here, this is just 10 examples of 10 different projects, 10 different tissues. We looked at 10 million cells, over 2 billion transcripts. Really a great tool that shows really the wide array of different research areas and different tissue types that we've worked with when it comes to NanoString, specifically around spatial biology. And all these can be found at a webinar repository that we have, which I'm happy to share following our talk here. And it's a great tool to be able to really look at not only what are the features around our platforms, but also really how they're being used and what are the data sets that are being generated. So really excited about that. And of course, as Adrienne mentioned, obviously our GeoMx digital spatial profiler, as well as our nCounter system, are located within the core and can be taken and used for running projects. So also, next thing I wanted to mention before kind of going into our technology and also thinking about some of the different case studies we have demonstrating our technology is I just wanted to share with you a recent publication that came out, which utilized our GeoMx digital spatial profiler platform. And this is where we used our whole transcriptome atlas assay that looked to construct high resolution molecular transcripts and spatial communities within pancreatic cancer for a study that came in Nature Genetics here earlier this month. So this is a great tool that really demonstrates being able to look at single nucleus and spatial transcriptome profiling of pancreatic cancer that looked at multicellular dynamics associated with neoadjuvant treatment. Again, this is found in Nature Genetics. So really exciting new publication that came out. Beautiful data set that you can see where we've imaged that particular sample type with our great profiling modalities that we have that are really unique to our digital spatial profiler. So first, just a quick recap of kind of how our technology is working and where we've started as a company, right? So we've got a long history of bringing innovative technologies to the market. So from our nCounter platform, which is a great mid-plex gene expression profiling system that does not require amplification. There's no conversion of RNA into cDNA. It's a great tool that sits right between qPCR and RNA sequencing, where we have a max of about 850 genes that we can profile for. And we continue to launch new products and new panels on this system, launching about 2 to 3 new panels every year. And the latest new panel that we're launching specific to nCounter is specific to our cell and gene therapy roadmap. So we have a gene therapy panel that's launching here later this month that really rounds out a great solution within immunotherapy as a whole. So great tool that we have there. And what folks really liked about nCounter was, one, it's simple, it's fast, it's easy to use, really great actionable data that you gain from that. But also, we work really well with FFPE, so really challenging sample types. And that was really kind of the creation of our company, was all around FFPE and being able to generate really meaningful data sets off of really tough sample types. And that's something that we've carried through with the rest of our platforms as we've continued to launch new systems. So again, we've been known for being really robust and reproducible across labs and a variety of different sample types. And we've really grown rapidly within the past 3 years. And much of our investment that we've since focused on for the past 3 years is really building out our commercial organization to really support the building demand around spatial biology products as a whole. And so we've come to recognize within spatial biology that there are multiple levels of structure, from macro tissues to individual single cells. And each of these require a different level of resolution to interrogate. So the level of plex that's required also varies with the question that the researchers are asking. So a 100 plex panel might be sufficient for looking at specific biomarkers or to perform high-level cell typing. And increasing that plex to thousands of genes allows researchers to interrogate differences in biochemical pathways. And a whole transcriptome approach enables an even broader discovery of novel biomarkers. And so this is where, if you're selecting spatial technologies, you need to consider both the level of plex as well as resolution and their specific applications that they require. So this is kind of gives you an idea of kind of where our technologies sit. So we've got whole transcriptome discovery within cellular substructures to pure cellular populations within genomics. So we've got glomeruli and tubules here. And then in parallel, when we're developing our CosMx platform, we're really looking to unlock the remaining insights that exist at single cell and even subcellular resolution. But with this, we're looking at about 1,000 plex assay as we are sitting at today. So we've devoted over $60 million in R&D spend and really developing the best-in-class solutions from our analysis. And really, all of our products benefit from the commitment to really 4 foundational elements, right? So superior performance, again, with an FFPE to ensure you get the most out of your data and from your most precious samples. We offer multi-omic readout in being able to look at the largest portfolio of optimized and validated content in terms of RNA and protein. We've also devoted a great deal into our automated solutions that really streamline our workflows, and those ensure us reproducibility across labs as well as users. As well as an integrated informatics pipeline that's very differentiated with the launch of our AtoMx platform, which we'll conclude our talk here with today, in demonstrating what AtoMx represents within spatial biology. So all these together have led us to a broad implementation of over 300 systems installed worldwide, and over 130 peer-reviewed publications that we have year to date as well. So also we're finding that flexibility is very important in looking at spatial biology. So from a genomics perspective, not only are we supporting a number of different tissue types, but we can also look at a number of different tissue formats as well. So we can look at a single section, we can look at multiple sections, we could look at tissue microarrays, right? Where our assay really goes in and it's differentiated, we aren't fitting the assay inside of the tissue. We're allowing the tissue to maintain its structural integrity and being able to look at all of these different compartments from a whole transcriptome perspective and over 2,000 different pathways that we can take a look at from our tissue. So also our assays are really well known for having a simple, standard histology workflow. So with our genomics system, we basically have some imaging reagents. These are 4 fluorescently labeled markers that help us to visualize our tissue. And then also we have profiling reagents. So if we're running our whole transcriptome assay, we're looking at the whole transcriptome, or we can also look at from a protein perspective. We've got over 150 protein markers that are utilized as well. And really this leverages standard histology, right? There's a standard positively charged slide that we use. There's no special slide you have to order. We maintain the histology workflows that are already well represented and well characterized. And we can take things a step further in being able to offer up some automation that we have. So also we provide great information just from a single slide, right? So we can look at over 2,000 different pathways. We can look at cell abundance scores. We can do protein and RNA. We also offer great scalable throughput and being able to profile up to 12 slides within a day with some of the automation that we have. And we can really work with any different sample type. So whether that be archival tissue from FFPE, fresh frozen, whole mounted, again, or even tumor microarrays. We also have a great panel offering of over 150 pre-validated proteins, and we'll share a little bit of what that panel content looks like. We also have an automated solution that allows us to actually go down to just under 30 minutes of hands-on time for preparation. So really a technology that can grow with users as they're getting up and going. So let's just talk a little bit about our flagship panel. So from a GeoMx perspective, this is our whole transcriptome atlas. So we've designed probes to every protein coding sequence within the transcriptome. So this has a non-poly assay. So it really creates robust performance across a variety of different RNA qualities. It also allows us to have great sensitivity in being able to detect both low, medium, and high expressing genes. There's also a customization option where you can spike in additional content if you want to look at non-coding sequences or even exogenous RNA content as well. There's minimal sequencing that's involved. And again, we also have a code detection assay where we can layer in protein, where we can look at proteogenomics with DSP. And here's a white paper that demonstrates our ability to be able to profile for both whole transcriptome. And in this case, we looked at over 147 different proteins from a single slide. And right, the benefit here is that we're actually taking and kind of linking transcription with translation. So creating a great deal of content by way of information you're gaining from those particular sample types. So here, in terms of sensitivity, right, we've got the best-in-class sensitivity. We've got over 5 logs of dynamic range that allows you, again, to detect both all low, medium, and high expressing targets from really challenging sample types like formalin-fixed, paraffin-embedded tissue. And here we demonstrate our high-expressing genes, our medium-expressing genes, and our low-expressing genes as well. So also consistency. So we have a wonderful partnership with a number of different platforms, one with Leica, where we've got an automated sample preparation workflow with the Leica Bond RX system. And essentially, this reduces hands-on time from 3.5 hours down to 30 minutes. And it also provides great slide-to-slide, user-to-user, and instrument-to-instrument reproducibility, and really opens the door for being able to do large cohort studies or multi-site studies as well. So here you can kind of look at our concordance we have between a variety of different sample types and gaining great reproducibility from that. So not losing any of the reproducibility you have as you turn to scale with the samples that you're working with within a given run. So here's our menu that we have with our GeoMx system. So it's a broad menu of pre-validated panels as well as custom options. So we've got the largest portfolio of protein content, and you can quantify over 100 protein targets simultaneously. There's modular designs looking at immunology, immuno-oncology, and even neuroscience research that allows for total flexibility within experimental control. So all our antibodies that we have are contained within this panel. They're validated together. So you don't have to waste valuable time or resources validating targets. Order the panel today, get it tomorrow, start running your experiments the next day. Major benefit there. We also do offer the option of being able to spike in custom targets as well as the ability to build out your own panels from scratch. So we have a protein labeling service here at NanoString where you can send your antibodies here and we'll label your antibody for you. We also have an antibody labeling kit where we can provide to you and you can label your antibodies as well. So we're creating great customization options you have from that perspective. From an RNA perspective, anyone who's familiar with running an nCounter assay, we've built custom nCounter assays for years. You just let us know your genes of interest that you want to profile for. We'll build it for you. We won't charge you a cost for building the assay, or we'll only charge you the cost for the probes themselves. And we offer great solutions of being able to spike that into whole transcriptome as well. So here we demonstrate, we have a COVID-19 spike in that we've utilized. There's a number of other applications from a cell and gene therapy perspective. We have a TCR diversity assay spike in that we're in the midst of developing as well. So a number of great solutions as we continue to look at providing custom solutions for as well. So we also got a great partnership with Illumina, where we've collaborated with Illumina to create this great analysis of GeoMx data that's read out on, of course, on an Illumina sequencer to get that whole transcriptome information. So for folks that have access to an XC 1000 or 2000, you can actually use that software on the GeoMx instrument, and that data automatically gets processed on Illumina's Dragon Bio IT platform. And then upon the completion of that sequence processing, the spatially resolved digital counts of that RNA or the protein quantitation that you're getting are then ported directly into the GeoMx system. And that allows researchers to interact, visualize, and analyze their multi-omic data sets. So we've also got a great solution. What GeoMx really enables for you to be able to simply accelerate your spatial discoveries with unrivaled speed and plex, right? Where you can quickly connect your tissue location with the function and regulation. So here we've got a great example of working with sample sets interested in glucagon and insulin. And we've got a great study looking at diabetes research with a case study here later on in the presentation that does just this from a GeoMx perspective, and then moves into it from a CosMx perspective of actually getting single cell resolution as well. So layering this in has become very important. So we provide great tools for automation that enables for large research studies and large cohort analysis tools with use of our Leica and our Illumina partnerships. And this has ultimately led to what we're now at over 145 peer-reviewed publications year to date using our GeoMx digital spatial profiler. So it's really, we've found that GeoMx has been a great use of being able to answer critically biologically relevant questions and providing across a variety of different research studies, certainly within immuno-oncology, within infectious disease, within neurology. And we've had a number of great covers that have come through using the GeoMx platform. So we talked about our GeoMx system here, and what we've also in the midst of launching is our CosMx system. And as a quick reminder, our GeoMx system enables whole transcriptome. It's got a great large dynamic range of over 5 logs of dynamic range, ultra high throughput, really a great tool for looking at these tissue structures. But we also found a need being able to get down to single cell resolution. And so each of our systems have an automated pipeline, are FFPE compatible, are multi-omic and being able to do RNA and protein analysis. And where our CosMx system comes in, it's really this high plex imaging across an entire tissue section. And where we're able to get into what we describe as really the foundation of single cell research and being able to do cell typing and mapping and being able to getting into more functional resolution of those particular sample types. So for our CosMx system, this is where we're able to get into single cell and even subcellular resolution. It's kind of a fun slide where we're thinking about CosMx and really what it really represents within the next chapter of single cell research, which is the largest plex of RNA and protein quantification and provides data analysis solutions that are really turnkey. So here's an example of the Hubble telescope, right, which was this massive resolution increase. It really changed the questions that could be asked and answered in astronomy, right? And we're already finding the same case as we're thinking about CosMx, where, from just a 5 micrometer thick melanoma FFPE sample, we're generating 1,000 plex RNA and looking at also 3 proteins for cell segmentation, getting this 10 terabyte size of raw data per sample. So just very rich data sets that we're able to generate from that. But also we have great flexibility comparable to our GeoMx system. As we built out our CosMx system, we wanted to certainly show that we had efficacy of being able to look at different sample types as well as cell type maps. And so essentially it's not just immuno-oncology, which certainly we are well positioned in being able to profile for, but also in many other different types of tissue types. So here we have FFPE from skin, from a core needle biopsy, we have organoids from brain, we have fresh frozen tissues from mouse brain, and also from tissue microarray from a lung sample here. And each of these dots represents a specific cell type. Just like with our GeoMx system, we offer great flexibility. So not only being able to profile a number of different tissue types, but a number of tissue formats, single section, multiple sections, or even getting into tissue microarrays where you're really getting into really rich data sets from that perspective. So really we've sought to create a single cell imager that really is used within the real world, right? So we've got a great, a very large install base of GeoMx current customers. And what we've really found is the importance of being able to just work off a common glass slide and having automatable tissue prep with common protocols for IHC, really a sample in, answer out design, and software tools that scale and really evolve for various project types and sizes. And that's what we've sought to create using our CosMx system. So for our CosMx system, it's a single cell spatial resolution system that addresses both discovery and translational research needs. So to date, we're launching 1,000 plex panel that you can also look at, 3 centimeter squared tissue areas of tunable throughput of looking at anywhere between 2 to 20 slides. It has 4 cartridge areas that can be profiled for within a given run. So we've got the highest plex and unbiased whole slide experiments that also supports really low plex analysis at higher throughput and any combination in between. So there's really maximum flexibility to really define your plex, your tissue and scan, and throughput based on different research requirements as well. So from our chemistry, it's a branched oligo dye fluorescent probe. So it's extremely bright. There's 16 to 60 dyes per probe. There's 4 dyes for 4 channels per cycle. And it's highly specific binding to different landing sites, which has become really well represented within NanoString as a whole. So for our standardized reagent cycling, it's really agnostic of panel type or targets. It's a universal cycle code sequence. There's 64 unique binding pairs, and there's 4 sets with each readout, and there's 1 for each channel or bit that's used there. So great solutions that we've had, that we've built out our CosMx chemistry. It was essentially, we've worked on this chemistry for about 5 years now. It was essentially first thought of, we were going to build a next-gen sequencer with an eye towards the clinic, something that was amplification free, no cDNA conversion, really simple, straightforward assay for that, and found a fit of being able to get into spatial biology, and found repurposing of this chemistry that has worked even better from a spatial biology perspective and getting to a really high plex. So great chemistry that's used for that. So here's the overall process for using or for running a CosMx experiment. And the subsequent slides kind of go through each of the steps more in depth. So this is really where we're really well positioned for looking at an industry-leading resource across each one of these areas, right? We've got different spatial publications that have referenced our being able to do spatial deconvolution, cell typing. We've got custom optics as well. So a number of great tools that really allow us to be able to get into the most informative information from your single cell spatial analysis. So first step that we go through, it's an automated low resolution scan. And it's really important when we're thinking about other platforms that would require a manual objective switching. So this is totally automated with the system. And this really helps to visualize and QC the initial focal assessment. So it's a first step that's automatically performed. And all these steps are essentially performed automatically on the platform. So next step was morphology markers and nuclei staining. So there's a multispectral imaging of 5 channels. And this really aids us in being able to run multi-analyte components. So within every RNA experiment, because we actually record a fluorescent signal map that can later be reintegrated into the transcriptionally derived data figures like a UMAP. And then the baseline of these is it's helpful for kind of selecting fields of view selection in certain cases with different cell types. So here we've used PanCK, CD3, and DAPI with that. So we also dedicate a channel to cell membrane identification, which is very, very important for being able to perform single cell analysis. So the majority of tissues, we use a cocktail of B2M and CD298, but it really works well for the ability to use others as well. There's certainly a customizable option of looking at different cell membrane features and tissues cocktails that we can use from a protein perspective to be able to ascertain the cell membrane staining as well. So one of the core competencies of our platform is really decoding the transcript barcodes at 1,000 plex to ascertain the ID and the localization and also the quantification of those particular genes per cell. So for the 1,000 plex RNA panel, we've got 16 imaging pools, and each of these are cycled into the tissue for imaging the 4 colors, and then it's got a PC quenched in the next pools represented here. So what we really look at is a multimodal approach when we're looking at cell segmentation. So we utilize AI and machine learning. So we have a cell pose assay that we utilize. There's customization to that as well, where you can actually look at other algorithms that you want to profile for. But we utilize a protein-based cell membrane marker for nuclei images, and then from a transcript-based segmentation refinement approach as well. So cell segmentation really is a multimodal approach that we utilize for the system as a whole. So also when we're kind of thinking about allocating the transcripts to single cells, right? This is where we're really getting into looking at XY positions and the IDs of these thousands of the RNA species that really total millions of transcripts mapped out to hundreds of thousands of cells that we're able to automatically perform within our system. And now we're kind of being able to perform the cell typing and mapping customization or features that's automatically generated within our system as well. So we're moving away from just what cells and how many to really the more biologically important questions of where, and depending on what we want to really profile for, we're able to look at low expressing targets or really rare abundant cells. And from this, we can actually look at, from this particular field of view, all these cell types that are essentially called out. And then we're able to do a 1,000-plex RNA quantitation piece out of that as well. So very rich and meaningful data sets. And so what we can then move into, and hopefully this graphic kind of comes through for us, this is the real reason for why CosMx is the next generation within single cell research, right? So we start at the endpoint of single cell RNA sequencing, a UMAP plot, and then place these clusters back into their biological architecture, right? And what that does is that gives us new findings on the contextual relationship, not only by positional density and composition or their neighborhoods, but by the intercellular signaling that's revealed on these 400 receptor ligand pairs that are profiled within our universal cell characterization panel. So really powerful data set. Really love looking at this, this graphic that kind of goes through and maps it back out. So we've also done some work around our protein assays as well. Of course, that's equally important here. This was from AACR 2022, where we actually looked at a 100-plex protein assay and being able to profile for that amongst the tissue as well. And then we have a listing here of our first manuscript that came out that essentially demonstrates the system performance. We introduced our CosMx chemistry, as well as some of the demonstrated applications. And then we have our first publicly available data set, which is a cell atlasing of non-small cell lung cancer from FFPE, where we profiled about 800,000 cells, 260 million transcripts. And what we like to demonstrate here, again, this is publicly available, but this really demonstrates our ability to work with real life samples, right? So these RIN scores that we have here, it's a DV200 linked, and they range between 20 to 60%, right? And this is where we're really working with real world samples with degraded RNA sample types. So next I wanted to go into a case study that we've generated that was a very interesting set that's from the St. Louis University School of Medicine. This is Grant Kolar, who runs a core facility there. He's a pathologist, and he utilizes both our GeoMx system as well as our nCounter platform, and is also in the midst of gaining a CosMx instrument as well. And so this is a great study looking at pancreatic islets of Langerhans that elucidates the metabolic dysfunction associated with type 2 diabetes. So I thought this was an interesting case study. I demonstrated our case study that looked at non-small cell lung cancer. So this is another data set looking at another different tissue type. And this is also, there's a webinar that's available for view as well, which I'll be happy to provide after our talk here today. So this was a great study where they were looking at the abundance and distribution of cell populations that are present within the islets of Langerhans and pancreatic tissue that were characterized within this. So they really wanted to look at the insight into ongoing immune response in the context of tissue damage. So this is spatially resolved, a single cell atlas of the human pancreatic islets. What this did was it provided really an unprecedented insight into the function of these endocrine microorgans in the context of health. And that provided valuable characterization of the molecular and cellular changes that are associated with tissue dysfunction as well as disease. So within this case, he used our 1,000-plex cell characterization panel. And from this, looked at all these particular cell types that were well represented and important within his study. And so from this, we were able to look at all these, about 1 million cells in total that they're profiled for, and we're able to pull out these particular cell types. And here we're looking at these specific endocrine cells that you're able to look at within this field of view that otherwise we weren't able to. And so what he really wanted to be able to look at was a spatial atlasing of these pancreatic islets in diabetic and non-diabetic patients. And so they were able to map subpopulations of the beta, the alpha, and the delta cells in a spatial context. And they determined the abundance and distribution of the subpopulations in normal controls and their predisposition in the diabetes patient population. So from this, they looked at 9 transplant donors based on age, race, and sex, and the ratio that was matched. 3 normal controls, 3 type 2 diabetes, 3 metabolically healthy obese, 3 pancreas rejected, but transplant either due to no recipient or based on visual inspection. And from this, again, they used the 1,000 plex panel plus the custom panel of 30 genes. So we can spike in additional 50 genes of your choosing into any one of the panels. And from a morphology marker perspective, he used the B2M CD298 antibody cocktail that we have that's off the shelf and readily available, including pan-CK. And he also looked at glucagon and insulin antibodies, so adding some customization for some important morphology markers that he wanted to have included as well. So nice customization that we can utilize. This was highlighted at AGBT this year, and it's poster 309 if there's interest in that as well. There's some more information on what that looks like. So again, Dr. Kolar is a pathologist by trade, and so he used an H&E stain to select the islets to kind of focus specifically, rather than just the whole pancreas. And for him, it was really important to avoid the digestive enzyme of these secreting acinar tissue. And so this really shows the value of being able to look at field of views within the tissue that can be equally important in pulling out these really rich data sets based on an H&E stain, and then also the morphology markers that we used as well. So a nice selection of tools to be able to help to indicate the field of view locations you want to profile for, and then what the system does with this low resolution scan on that first step of the process with the instrument. So we talked a little bit about the panels. So just to kind of reiterate what we currently have available. So we have our predefined panels. So this is a human universal cell characterization panel. The next slide I share with has the information of their annotations. And we're happy to provide the Excel sheet that provides that as well, if that's helpful for folks as they're thinking about projects and how that could be utilized. Again, this is a 1,000 plex assay. It's the highest plex single cell spatial assay that's available. We also have a smaller 100 plex assay that's focused on immuno-oncology. Just launching our mouse neuroscience 1,000 plex RNA panel, and also our 64 plex human immuno-oncology panel as well, with more panels following suit from a mouse perspective as we're getting into the new year as well. So again, we can do a customization. You can spike in as well as the standalone custom options. So you could spike in an additional 7 to 50 genes of your choosing into the 1,000 plex panel, or 7 to 10 into the 100 plex RNA panel, or there's an option to do customization for the protein perspective as well. Or if you're just finding that you want to profile for just the genes of interest that you want to profile for, no problem. We can do that. We can design up to 300 genes today. More to come, more than likely. But we can profile up to 300 genes today of your choosing. And that's a major strength for NanoString and being able to create custom assays. Again, we've made over, I think the number we're up to is over 10,000 custom code sets on from an nCounter design perspective. And so from a bioinformatics perspective, we've got a great history of building out really great assays from a total custom perspective. You just provide us with the gene list and we can profile for that. Our chemistry allows us to be able to look at lots of different gene sets that many other platforms cannot, because they could perhaps be too short in length. We have a great way of being able to look at lots of different genes. So good solutions that are there. So from a cell characterization panel perspective, this is the off-the-shelf system that we have that's really geared towards our panel that we have that's really geared towards looking at cell typing and cell-to-cell interaction studies. So the gene sets that we have go everywhere from looking at cell typing and mapping, these cell-to-cell interactions. There's a ligand receptor family piece that's included as well. It's very differentiated, as well as all these other different signaling pathways. And we also go into our cell state and function breakout and being able to profile for all these, from wound healing, hypoxia, cytotoxicity, being able to profile for cytokines and some really challenging sample types as well is one of the major strengths of the platform. And here's also what we have by way of our 100 plex panel looking at really immuno-oncology. So looking at key immune and stromal cells that we have that are very mission critical within these types of studies. Again, it's a great way of being able to look at the immune cell types, their activities, the aspects of the tumor biology that's relevant within IO studies. So all these different cell types are automatically included, from fibroblasts to B cells, and also the IO physiological traits as well. So looking at cytokine and chemokine signaling, which is, again, is the strength of the platform, looking at these short transcripts in length, and also getting into cell cycling as well. So all of our assays do also include negative controls as well as a means of QC and understanding the success of those studies as well. From a neuroscience panel perspective, we've got our 1000-plex panel that looks at 6 key themes, so 44 different pathways. So we're finding many targets, including glial activity, neuropathology, cell stress damage, neurotransmission, inflammation, all these areas that are automatically included. It's a larger plex panel, so it does include these 10 negative controls as well. A great tool for being able to look at robust cell typing as well. This just gives you a little bit around, as we're thinking about custom RNA targets and just kind of the details of my formatting here. Got a little off here, but when we're thinking about doing a custom add-on where you're adding on additional targets, essentially, again, we can do up to 50 genes of your choosing into the 1000-plex panel, and we just need to know what those targets are, and we can build those out. We typically build sizes about 192 slides worth of increment with custom options that are built for that, and that's true whether or not you're adding that into the 1000-plex panel or you're going with the standalone panel as well. So good solutions there. This kind of gives you an idea with our immuno-oncology protein panel content that we have. So we've got this word graphic that kind of demonstrates where we've put the majority of the different protein targets that are there. So checkpoint, myeloid, which we import with immuno-oncology, but really it's a great immune profiling panel. So looking at cancer, stroma, and controls. And we include all these different protein markers that are included. So lots of different options we have there. So that's our kind of our panel offering and kind of an overarching thought around what CosMx is, how the technology works, and a data set looking at diabetes specifically that I hope was impactful there. I next wanted to share a little bit around AtoMx. And AtoMx is something we're really excited about because this is a solution that we feel really helps us within the spatial biology ecosystem, right? AtoMx is a platform, it's a spatial informatics platform that will allow us to be able to incorporate genomics profiling studies, as well as CosMx imaging studies. And there's talks about being able to incorporate meta-analysis components as well. So looking at other platforms and combining other data sets into that as well. But as we launch AtoMx, it's going to be launching with CosMx. They will be equally important in being able to import data into the cloud and run our CosMx data sets through AtoMx. And AtoMx will enable us to be able to perform image analysis, data visualizations. There'll be support for machine learning as well as AI. It will help with storage and providing really scalable storage solutions as well. As well as a great tool for being able to get into global collaboration and being able to share data sets amongst colleagues as well. And there's a nice feature of kind of what we're building the system out is it's got a great architecture and being able to look at really scalable architecture, standardized data formats, which I'll share a little bit of what those look like. There's an integrated analytics that goes through from GeoMx and CosMx. And we're also building out third-party and open source tools as well. So building out for region of interest selections and using AI algorithms for that, open source analytics, digital pathology analysis, and spatial AI. It really helps us to build today and be able to work with GeoMx data sets as well as CosMx data sets, but really gives us an idea to the future of being able to incorporate lots of other data sets as well. Really getting us ready for the cloud, third-party analytics and open source or even custom modules. So next, I just wanted to share a little bit about this integrated workspace that we have that manages studies and images and results across GeoMx as well as CosMx. And so this is a tool where it really offers scalable storage and direct access for all your experimental data. There's a streamlined analysis tool and pre-configured and customizable pipelines, which I'll share in a couple of slides here. And then again, you can really share your data sets with collaborators around the world here. So this is our pipeline orchestrator that really provides pre-configured as well as customizable pipelines. So with any course of this experiment, right, you could include and introduce a customizable pipeline that you can incorporate into this pre-configured data set. And so there's great flexibility that we have in being able to maintain that customizable pipeline that's there. We do also offer a data analysis service. So today, for any GeoMx assay that you're running, there's a cost that's associated, but you can actually work with our bioinformatics team, and we can actually serve as your bioinformatician in looking at initial data sets or building out a pipeline for a study that's going to be reoccurring, and kind of then incorporating that into AtoMx is something that we feel like is a major differentiator as well, being able to work with data sets in that way. So here we're demonstrating a little bit around these modules that we built out, where we really feel like this is a best-in-class data analysis and visualization program that we have with AtoMx. So here we have our foundational modules. So anyone who's running a spatial analysis assay, you're going to want to QC, normalize, having access to UMAP, being able to perform the cell typing, PCA, and getting into basic spatial clustering. And then also kind of getting into these spatial analysis modules, where because we have the highest plex assay, we have this great ligand receptor offering within our panel for the cell characterization panel. And then we can start getting into nearest neighborhood and spatial network and cell proximity analysis, as well as differential expression tools as well. We have pre-configured pipelines specific to RNA or these foundational pipelines for protein, or we're also designing assays that allow us to more get into these custom modules that are really user-defined as well, as we're moving into areas for that. So I wanted to be able to have enough time to be able to have some discussion if there's questions that folks do have. This is the last slide that I have. So this is, again, the great webinar series that we have that's coming out. It's the first one we have is with Jake Estes at OHSU, who has a focus around infectious disease, specifically looking at lymph node, utilized our assay for an HIV application. And then the case study that we presented before with Grant Kolar has a diabetes focus, looking at pancreas, and a number of other great studies kind of local kind of to the area as well. Pramber Dulai out of Northwestern has a great study looking at GeoMx and CosMx data, looking at IBD, kind of bugs in space type of an application, looking from colon. And then also Andrew Roth from UPenn has a CAR T cell application, which is really interesting because he's incorporating nCounter data sets for cell and gene therapy and using the CAR T panel that we have there, and also using our TCR diversity analysis tool that we have from our GeoMx platform as well. So lots of great studies, lots of great application areas. We're really excited about it. I'm very appreciative of the time here. Maybe I'll just kind of look here and look at a couple questions that we have here. So one of the questions here is a fee-based service to initiate a research project in order to get preliminary data for grant applications. So it's a great question. So I think of that in two ways. So when it comes to GeoMx, right, there is a system that's located through the Stark Neuroscience Research Institute, including the nCounter. That's a great tool and resource to be able to start there, perhaps. And we can work with you in understanding it's your first project. We've got a local team that's always eager to help and talk about your project, the goals that you have, understanding what the spatial question is, understanding how we can help you to really generate meaningful data sets. So that's something that is one idea. We can always, work with you and kind of work with the core as well, and being able to offer a really nice solution for your first experience there. We also, from a CosMx perspective, that system, we do have a program called the Technology Access Program, and the Technology Access Program does allow the use of being able to generate initial data sets with our technology access program. So essentially, we'd be able to come up with an idea of like, what are the spatial questions that you have, and what are you wanting to kind of gain from that experiment. And so we would have a kickoff call with our scientists, and we'd be able to understand what the project sample types that you're using, utilizing our cell characterization panel. And then we can actually use our team in-house to be able to serve as your technician, and then also with your bioinformatics as well. So for the CosMx perspective, we can run a handful of samples and running that as well. And if there's interest in doing that, we'd be really happy to be able to work with any data sets and from those types of projects. There is, so Luke has a question around the general guidelines for kind of powering a single cell spatial experiment using the system, like number of cells, but also biological samples. There's a great, I don't have it here, but there's a great white paper that actually goes through with experimental design that I'm happy to share with the team as well, that just kind of talks about, thinking about the number of samples that are going to be used, kind of what the spatial question is that you're wanting to get answered to. But I'd be happy to share that slide that we have that's around that as well. That's a great question. So cell characterization panels for mouse and rat. So on our product horizon, so we have human and mouse. So mouse certainly will have a 1,000 plex mouse cell characterization panel that'll come out as well. We're also really interested in just hearing, what researchers have of interest as well. So there is a nice opportunity, we feel like, with CosMx to be able to do consortium type builds as well, because the consideration is always, whether or not they want to, our folks in-house want to be able to fully commercialize and market a panel and sell it on our website. Whereas if there are like-minded folks and researchers who have common genes of interest that they want to profile for, and we could design them specific to rat, for example, those are things that we're very interested in hearing about. So if there's an idea of a particular sample type, and if it perhaps spans across a couple different collaborators, there's always a means of building that. But we can always do the custom route as well. That's always something we can profile for. And I think so there's a CosMx that's there as well. Yeah, so the protein expression, it is quantifiable. So it does provide quantitation of that particular protein or antibody that's present within the sample set. So it does, and or a list of validated antibodies. We have a listing of yes. So from the GeoMx system, we do have a listing of the specific proteins we have of interest, or that we have included within that. Let me see if I have that within this presentation, or I can try. Happy to provide it to you guys. But it does provide all of the specific proteins that we have. So there's over 150 that are available. And then those are pre-validated, but we also can build protein assays specific to your proteins of interest as well. Agreed. So this the creation of different panels for dogs and cats. So we have a companion, it's a companion oncology panel that we first designed for the nCounter that's specific to canine. And then what we did was we found great interest from a spatial perspective. So we've since built, in the process, I think in September, we're going to be launching a canine oncology panel for GeoMx. So I'm happy to keep that down, and I can provide you with the gene list, what that looks like and when that's going to be coming out. But that is a panel that we're in the midst of launching. So I know we have that for dog. I don't know if we have anything on the horizon for cat specifically, but I think it's always good to hear what the interest is and what we can build out as well. So that would be good to hear, but I'm happy to provide the listing we have for dog. Yep. So just if you're just curious about the cost of the instrument for the genomics system, we've already got that located here in the Stark Neuroscience Research Institute, but that runs right under. It's at $295,000 for that system. Yeah, I think. I think so. When we're kind of thinking about our CosMx platform, and we're thinking about other platforms, what CosMx represents. One, this is the second spatial system that we've built out, so we really have the team and the infrastructure to be able to support a product launch, to be able to support those systems, to install them, to be able to really have the right type of engineering team and scientists to be able to support those systems being launched. I think that's a major benefit of our platform. We offer the largest plex, so we offer 1,000 plex panel, and that plex will grow. We have line of sight of being able to demonstrate our ability to do that. There's no conversion, there's no amplification that's required with our platform either. There's no tissue clearing. In terms of our workflow, it's really a major strength is being able to go, it's about an hour's worth of hands-on time, and a day's worth of kind of processing. So what we're doing is we're providing with a really streamlined workflow where we've already demonstrated our ability to work very well with IHC applications and being able to profile for that. And also what we can do is both RNA and protein. We can do that today, right? And all these case studies, they demonstrate everything that we've already done with CosMx. So I think that there's a major benefit with where we are within the single cell imager marketplace is we're already here. We've already been generating data. We're already working with customers. We've already demonstrated how our technologies work together with other platforms as well. And I think that our ability to be able to continue to offer really nice solutions and continued work around single cell imaging, there's a major investment that we've made, and that's something that we'll continue to do as well. I'd also look at our Atomics platform. I think that's a major benefit, a major strength compared to any other platform that's out there. And we have a lot of information around CosMx already. So major points of difference, and the Plex is going to just continue to grow, and we'll be able to continue to generate very, very meaningful data sets from that large Plex. Some would think that there's a trade-off with offering a higher Plex assay, and there being a longer kind of bioinformatics pipeline that you would have there. We would actually argue that actually speaks to our being able to provide really actionable data because of the Plex, and are having a really nice solution with Atomics as well. So really important. Lots of interest within dog and swine. Okay, that's great. I mean, so I think that maybe what we can do is we can kind of demonstrate to the team what we're doing from a dog perspective. We also have a lot of folks who use our whole transcriptome assay from a genomics perspective. And we have great cross-hide studies, like for swine, for example. There's a number of studies that use our whole transcriptome for human and find that overlap is pretty nice. So I'm happy to provide kind of a cross-hide list of working with porcine using our human panel. That could be of interest. I'm happy to send that over to you, Abby, if that's helpful. Spatial transcriptomics with pancreatic tissue. Yeah, so we do have case studies of kind of working with the pancreas. And this is one study what Grant Kolar has actually demonstrated. He said if there's ever interest in talking with Grant from that kind of perspective, he's wonderful to work with, great collaborator to be able to talk with folks, loves to talk projects and science as well. But there is a great tool of being able to really profile on the right types of cells, right? We can profile for those cells types that are specific to the pancreas, so we don't have to worry about concerns of RNases. So that's one of the major benefits of the platform, and being able to profile in that way. But happy to demonstrate a little bit more on what that looks like as well, if that's helpful.
So thank you, Seth. If there's anyone else that would like to unmute themselves and ask a question, yeah, feel free to do so. We have a few minutes remaining.
This is Adrienne again. So you can feel free to email biocore@iu.edu and we can get you in touch with NanoString if possible and give you the prices. So we do run for a fee for service, and there is a discount for academic institutes compared to external pharma companies. So feel free to email us. We're always really responsive and we'll be excited to work with you. I just saw the CosMx. There's been discussion, Jill, about purchasing at the CosMx. So email us. Certainly, we'll let you know if there's any updates.
Seems like there would be great interest in that.
Yeah. Thank you, Seth.
Thank you so much for the time. I really appreciate it. Thank you so much for the questions, and I really appreciate it. So thank you again for the time.